Yeah. So thanks for the introduction, Sonny. As you said, I'm Matthew Peill. I'm the molecular biologist with Perennia's Plant Health Lab. And I'm going to be talking today about managing aphids and viruses in raspberries and strawberries. Sort of giving an overview of what viruses and vectors we have in Nova Scotia, as well as some of the work that we've been doing, developing testing protocols in the lab, and looking at management practices within Nova Scotia and how they may be influencing viral disease development. So I'm going to start off with a brief history. I imagine most people on this call today are going to be familiar with this, but viruses started being a big issue in Nova Scotia back in 2012 when there was a major outbreak of strawberry decline disease. So strawberry decline disease is a general name given to virus diseases in strawberries, generally referring to when you have this massive decline of plants caused by complexes of two or more viruses. Generally speaking, single infections aren't going to cause this level of disease, and you need those two or more viruses to cause an issue. The picture actually on the right is a field, or a picture of a field taken from that time, where we had essentially a 100% infection rate. But the symptoms that you see, the reddening leaves, the stunted plants, and sort of general decline is what you would expect to see in a serious case of these diseases. So in response to this, there was a bunch of testing done in the following year of 2013. There was high virus levels and vector levels found within the province, and the majority of fruiting fields were destroyed to help prevent the spread of virus. There was also impact to the nursery industry in Nova Scotia, and it was millions of dollars of losses for the industry as a whole. In response to this, there was a provincial strawberry virus monitoring program launched, and within that, they also looked at raspberry viruses and vectors, as raspberries, which are closely related to strawberries, are susceptible to similar virus complexes. So I'm going to start with the general overview of plant viruses. I mean, now with the pandemic and things like that, most people are familiar with viruses, and some of those general traits cross over into plants, but just want to give a general primer on them so that we're all on the same page. So viruses, unlike most of the pests that affect crops, are non-living, or considered to be non-living. They can't replicate outside of their plant host. They're considered obligate internal parasites. So they require the cellular machinery of their host plant to replicate and cause disease. So you're not going to find virus sort of free living in the environment, and they're only going to exist inside of the plants they infect. So they exist as these particles, which are shown to the right. An individual virus particle is referred to as a virion. They're very, very small. So this is a graphic of one, obviously computer generated, but the image below is actually a transmission electron microscope image of viruses extracted and purified from a plant. And they're about 10 to 20 nanometers in length, or about like 1/10,000,000th of a centimeter. So very, very tiny. Obviously not something that you can see with the naked eye. And even when they infect a plant, they can often be mistaken for other disorders and diseases. Because of this, they're considered cryptic, and generally you need molecular or serological testing techniques to confirm diagnosis. Also, once a plant is infected in the field, there's no way to eliminate the viral infection from that plant. So once you have the virus in that plant, the only thing you can do is remove that plant to get rid of the infection in the field. And unfortunately, there is no way of directly controlling viruses with conventional pesticides. So most of the management of viruses is based on controlling the ways that viruses move from plant to plant and removing those reservoirs of viruses, such as infected plants. So most people are familiar with the plant disease Triangle, I've modified it slightly because things are a bit different for viral diseases. So instead of environmental conditions, generally the main way that, or the way that viral diseases are spread are via vectors, or plant viruses specifically, or via vectors. So as most people are familiar with, most of the viruses that we have in Nova Scotia, the vector is aphids, but there are a variety of different vectors of plant viruses, nematodes, pollen is another way. But like I said, aphids is the main one of concern in Nova Scotia. Susceptible host plant is another component of this triangle. Most or all of the strawberry and raspberry varieties that we grow in Nova Scotia are susceptible to the viruses that we have. There's varying degrees of susceptibility of those varieties, but all of them can be infected with virus. And then the final component of the triangle is inoculum, which is the infected plants, either commercial fields, or there's also wild host plants that are susceptible to these viral diseases as well in the environment. So there are many strawberry and raspberry viruses, but there's only two each of concern for each strawberry and raspberries in Nova Scotia. So the two main viruses of concern for strawberries are strawberry mild yellow edge virus and strawberry mottle virus. And for raspberry viruses, it's raspberry leaf mottle virus and rubus yellow net virus. So for both of these viruses, or for all of these viruses, single infections can be asymptomatic. In strawberry specifically, generally single infections is not going to cause disease. This varies a little bit with raspberries, where you can have disease develop with single infections of either raspberry leaf mottle or rubus yellow net virus. But generally, you're not going to have those serious outbreaks of disease unless you have multiple infections. But even at that point, these symptoms can often be mistaken for other diseases and disorders. And all of the viruses that we have in Nova Scotia are vectored by aphids. So the primary aphid vectors of the strawberry viruses are strawberry aphid, Ketosyphon fragifoli, pictured top right. These guys are generally pretty easily identified by the little hairs on their back. Obviously, you won't be able to see that without a lens, but it's generally pretty easy to see if you have one of those. And they're about 1 to 1.5 millimeters in length. And in raspberries, the primary aphid vector is American large raspberry aphid, Amphorophora agathonica. It's pictured in the bottom right. This aphid is quite a bit larger than the strawberry aphid. It's usually around 3.5 mils to 4 millimeters, and it's, other than the fact that it's one of the larger aphids you'll see, one of the dead giveaways for it is the sort of distal swelling of this structure known as a siphuncuли on its back end. So with both of these aphids, the way that they spread viruses is the same, though they only spread the viruses of their respective crop. So they feed on the phloem of either the strawberry or raspberry plants using a stylet, which is a feeding tube. So they pierce the vascular tissue of the plant, feed on the sap, take up the virus particles that we were talking about earlier. That enters into either on the stylet or partway through their digestive system or in their into their gut. And then when they move from plant to plant, they carry those virus particles with them and can then transfer it to another plant when they feed on it again. So aphids will have two different reproductive pathways. So during the summer, they will reproduce asexually and give live birth to aphids. If you look in the bottom right of the large raspberry aphids, these two little dots right here are actually the eyes of a baby aphid. You can see quite a few of them in the abdomen. So basically, these guys, once they are on the plant during the spring, become essentially baby making machines. For the strawberry aphid, they have about a 4-week lifespan, and during that period, they can produce up to 40 live offspring. And when they're giving birth to more aphids, those aphids, when born, can be already, the aphids, before they are born, can already be gestating more aphids. So basically, once they're born, they're ready to start cranking out even more aphids. And because of this, you can very quickly have exponential population growth if you're not managing aphids in the crop. Additionally, you have winged and non-winged morphs, and the winged morphs of the aphid are the most important in terms of spreading viral diseases to new uninfected fields and also aphid populations. So this diagram here kind of summarizes what I was just talking about. So I should say the second pathway for aphids is generally in the fall. They will transition into egg laying. These eggs are laid in the crop and are cold tolerant, or cold resistant. So these eggs are laid in the crop, and then aphids will hatch out of them in the spring. And once you have those aphids hatch out, they will start that exponential growth phase of the population and start feeding on the plants. If you have virus in the field, you can have the aphids feed on the virus and then start transferring that virus to other plants very quickly in the season, or very early in the season. As the population grows, or if plant quality decreases, you'll start to get the production of those winged morphs. In strawberry aphids, generally we see a flight period in mid-June to early July, and that's generally the major period where we start to see aphid populations spread and have the highest risk of virus being spread to new uninfected fields. So aphids are poor flyers, but once you have the development of those winged morphs, they can move short distances of a few meters within crop, or they can get caught up in higher level wind movements and be transferred several kilometers to new fields. They're generally considered poor flyers, so they can't really control where they go, but they can get picked up in that wind column and transferred over those areas. And once they perceive a strawberry field, they drop out of the column, and then they can fall onto the strawberry plants and start transferring virus and building up populations in those new fields. And then once again, transitions back to egg laying. And although this is showing, or this diagram is for strawberry aphids and strawberry viruses, it's a very similar process for raspberry aphids and raspberries. So another component of viral disease development within Nova Scotia is wild hosts. So obviously wild strawberries, wild raspberries, wild blackberries are very common. And they are known hosts for the respective aphid species. So wild strawberries, you will find strawberry aphid on those, and large raspberry aphid you can find on raspberry and blackberry species. There is also potentilla or cinquefoil, which is very closely related to strawberry and is known to be is a known host of strawberry aphid as well, but it hasn't been established whether it can actually host strawberry viruses or not, or hasn't been displayed to host strawberry viruses in the wild. And similarly, rose species can be a alternative host for strawberry aphid, but once again, it hasn't been demonstrated in the wild that they are a reservoir for strawberry viruses. So the main species of concern with this are your wild strawberry and wild raspberry and blackberry for their respective viruses, but there's also potential for potentilla and rose species to be hosting both aphids and viruses in the environment. So in terms of the current status of viruses in Nova Scotia, the Virus and Aphid Monitoring Project concluded in 2020. Following the destruction of infected fields in 2013, there was relatively low levels of virus and aphid populations from 2014 to 2017. But we began to see a rebound in these levels 2018 onward. And since 2020, outside of this program, there hasn't been any widespread sampling within Nova Scotia to look at virus levels. So at this point, we can assume that those trends continue, but we don't really have an idea of where virus levels are at, other than they were returning to higher levels. Part of the reason we assume that this is happening is that management practices on farm are likely contributing to this, either not as strict as management as or for aphids as there was during the virus outbreak period. And then one of the other issues within the province is that there is no testing service within the province for viruses, and we have been sending all of our samples out to labs in other provinces. And these two graphs here kind of show that rebound that I was talking about. So the graph on the left is the percentage of total samples that were positive for either strawberry mild yellow edge virus or strawberry mottle virus in each year of the program. So obviously the highest levels were in 2013. You start to see this decrease through 2014 to 2017, and then a rebound through 2018 to 2020. And the graph to the right is showing essentially the same thing, but it's instead of showing the total number of positive or percentage of positive samples, it's showing the percentage of blocks infected with both strawberry mild yellow edge virus and strawberry mottle virus. So in both cases, see that decrease and then increase again through 2018 to 2020. And then this is the same graph as the one on the right, but once again, showing that U shape, but then also a general trend of aphid populations increasing over time, which is the black line right here. So in response to this, Perennia, in partnership with Horticulture Nova Scotia, launched a small fruit virus project, trying to look at management practices and how they affect viral disease development in strawberry and raspberries, and also to develop testing capacities for virus in Nova Scotia. So the major objectives: determine the effective management practices on viral disease development. Also, while doing that, look at the levels of viruses in wild host plants growing in the borders of strawberry and raspberry fields, and then develop and validate small fruit virus testing protocols for Perennia's plant health lab. So to do this, we selected 8 sites, 6 strawberry sites, 4 fruiting and 2 nursery, and then 2 raspberry nursery sites. For looking at viral disease development in the field, we did a slightly different testing protocol compared to or slightly different sampling protocol than what had previously been done in the province. Generally, blocks had been sampled before by just collecting a random sample of 60 leaves by doing diagonal passes through the field. For this specific study, we did a more systematic approach where we selected 20 sampling areas within the field, each of them composed of 5 plants each. And these sampling areas were sampled each time that we sampled the fields that we were looking at to track disease development. So in this case, instead of collecting the 60 random leaves, we would be able to accurately determine the increase of virus in the field, or more accurately track the development of viral disease in the field compared to the other method. And then for the wild host sampling, we identified wild host species in the border of the fields that we were sampling from, collected those samples, and then tested them for viruses. And all of this testing was done by Perennia's Plant Health Lab using PCR, which is the standard molecular technique for testing for these viruses. So I'm mainly going to focus on the fruiting strawberry sites as part of the study today. So initially we were supposed to have 4 fruiting sites as part of the study, but one was removed in the second year due to poor establishment. We chose sites that were in their establishment year in 2021, which was the first year of this study to track viral disease development. So basically in the planting year, we did virus testing, and then we did follow-up testing in the following year in 2022. And for all of these sites, we collected information on the management practices used in these fields via survey to correlate them with virus infection levels at each site. So for the 3 sites that we looked at, there was a variety of different factors that could be contributing to the virus levels, but I just wanted to point out one in particular. So you'll see from the graphs here that site 1 had the highest levels of viral disease, whereas site 2 had relatively low levels, and there were no viral infections at site 3. One of the major differences between these 3 sites is that site 1 had that site 2 and 3 had monitoring programs, whereas site 1 did not. And this is monitoring programs for strawberry aphid, sort of illustrating the importance of monitoring aphid populations in the field to prevent viral spread. So in the first year of the study, there was a 70% infection rate of strawberry mild yellow edge virus at site 1, and 5% infection rate of both viruses. So one sample came back with both. And then we see this increase in the second year. So the overall number of single strawberry mild yellow edge virus infections decreases slightly, but that's only because there has been an increase in the number of co-infections of strawberry mild yellow edge virus and strawberry mottle virus. So in total, from the first year, there was a 75% infection rate in the field of at least one virus, increasing to 90% in the second year. And with that 35% double infection, you would expect to start seeing strawberry decline disease symptoms in that field, reduction of yield, visible symptoms in the plants, things of that nature. Whereas with site 2 and site 1, site 2, relatively low levels of virus, and it maintained the same from the first to second year. And obviously in site 3, there was no virus picked up at all. And this also aligns with some of the results that we got from the initial virus monitoring program. So both diagrams to the right, the top is for strawberry mild yellow edge and the bottom is for strawberry mottle virus. Shows the differences in virus infection rates based on sites that had monitoring programs and those that did not. Although it's not majorly or not super large differences, you were generally seeing less virus infections in those farms that had monitoring programs as opposed to those that did not. Once again, illustrating that need for a proper aphid management, aphid monitoring program on your farm to make sure that you're not having the spread of viral diseases. So I just want to touch on aphid management since we were just talking about it with that. Like I said before, monitoring is crucially important for effective aphid management. If you don't know if you have aphid viruses, or sorry, not aphid viruses, but aphids in your field, you can't control for them effectively. Aphid management is particularly important in the establishment year of fields. If you can prevent that establishment of aphid colonies in the field and that carryover of overwintering eggs in the field, you can prevent that early buildup of aphid populations in the fruiting year of your field and only have to worry about flight periods at that point. So in the first year, your biggest concerns are introductions of aphids into your field during flight periods. Like I said, mid-June to early July. And then again, there's usually one in the fall. And also if you have existing aphid issues on your farm, the potential movement of aphids between fields. Generally, the best way to prevent this is practice biosecurity between fields, move from new fields to old fields to prevent that introduction. And so preventing that establishment will hopefully prevent you from having overwintering populations in the field and that potential for high populations developing very quickly in the field from those overwintering populations. If aphids are established in the field, control should be used as soon as possible in the spring. And if you do not have aphid populations established, you can wait until your scouting service either finds aphids or you can apply controls once there is reports of flights in your area. So now I'm just going to move on to talking about the wild host testing. So over the 2 years of the project, we collected 109 wild host samples and tested them for all 4 of the strawberry and raspberry viruses. As I mentioned before, potentilla or cinquefoil and roses had sort of been hypothesized as a potential reservoir of virus in the environment. But from all of our testing, they came back negative for all of the viruses we were testing for. Additionally, strawberry viruses were only present in the wild strawberry species that we tested, and raspberry viruses were only present in the wild raspberry samples that we tested. So this is just a quick graph showing the virus levels that we found in the wild plants that we sampled. So we only found strawberry mild yellow edge virus from the strawberry samples. There was no strawberry mottle virus. And about 1/3 of the wild strawberry samples that we tested came back positive for that virus. And we've known, or they've known since the 60s that, or I mean, pretty much forever, that the wild strawberries have been hosting these viruses in the environment. It's a thing to keep in mind if you see these populations growing on the side of your fields. It's potentially a reservoir of virus that could be, or that could reintroduce virus into a newly planted field outside of the commercial field or an infected commercial field. And we had a little bit more variety in the rubus species. So we found both raspberry leaf mottle virus and rubus yellow gnat virus in the wild samples that we tested. So there's actually 4 species of rubus that we found. So wild blackberry is this guy right here, and wild raspberry is this column to the far right. This particular species, Rubus hispidus, is a ground growing raspberry species. It's a bit viney. You probably have seen it in proximity to the field, but it doesn't appear that it is a major concern for raspberry viruses in the environment based on the results that we got back, and based on its growing and how close it is to the ground, it doesn't appear that it's a good host for the raspberry aphid. And then we also have the Canadian blackberry here, which is, for most people, probably indistinguishable from a standard wild blackberry, but we separated out for the purposes of the study. But in terms of practical management in raspberry fields, really you should be looking to just control those raspberry and blackberry brambles in the perimeter, because they are harboring viruses that could potentially be transmitted back into the field. And especially for raspberries, just under 50% of the samples that we tested were positive for at least one virus, and we had a high degree of double positives for both raspberry leaf mottle virus and rubus yellow net. Slightly less than blackberry, but still concerning levels of virus in those sources outside of production fields. Last, I just wanted to talk about the virus protocol or virus testing protocol development within the lab. So as part of the project, we were developing protocol to test for virus in the Perennia plant health lab. So before we actually went through and did the project, we developed these protocols and validated them by sending split samples to an external lab to look at or to double check to make sure that we were correctly diagnosing the samples that we collected, and as well as to confirm positive controls. This all went very smoothly. And now the lab is able to offer testing for both rubus yellow net virus, raspberry leaf mottle virus, strawberry mottled yellow edge virus, and strawberry mottle virus via PCR. And then we are also able to do ELISA testing for strawberry mottled yellow edge virus. We're only able to offer strawberry mottled yellow edge virus via ELISA because there are not ELISA tests developed for the other viruses. But there are other ELISA tests out there for other viruses, and it's potentially something we can look at doing. So I just want to finish off here shortly by going through just some best practices in general for virus management. So outside of monitoring for aphid populations, I cannot emphasize enough the importance of planting certified virus-free stock. If you are not planting virus-free stock, you're starting on the back foot in terms of controlling viral disease development in the field. Generally speaking, this is something that's been put forward by sort of subject matter experts like Dr. Bob Martin, who was based out of the States, but you're basically looking, or single virus infections in the field aren't generally going to cause disease, and that's fine, but you're really trying to prevent that buildup of double infections in the field, which are going to cause your diminishing yields and obviously poor plant performance. So having any of those introductions early by not planting virus-free stock is going to seriously affect the longevity of your field, or the potential to carry it forward for multiple years. though generally speaking, at least for strawberries, it's not recommended to carry over a fruiting field for more than 2 years. So I guess 3 years in total, including establishment, but then only 2 years of fruiting. After that point, the risk of viral disease development is too high, and there will be diminishing returns on the field. And once those fields are in their final harvest, they should be removed as soon as possible after that point to prevent any potential aphids or viruses from leaving those fields, and also control the wild hosts in the border of those fields. Specifically with that one, you're trying to remove as much inoculum as possible to safeguard your new plantings. Also in the same vein, rotate placement of newly planted fields so that they're not close to those potential environmental sources of virus that could have built up with virus while the field was previously in that field. And then, if possible, isolate plantings from each other so that there's less of a possibility of aphids and virus moving from older fields to your new fields. And if you are holding fields over for extended periods of time, do testing on your farm to get an idea of where your virus level is at. If you have high levels of virus in the field, consider removing it, as it can have impacts on your farm, surrounding farms, and will generally produce poorer returns than replanting it. So just in summary, small fruit viruses are still a concern in Nova Scotia, and continued monitoring and management is needed to prevent major outbreaks from occurring again. To reiterate the points that I was just going through, start clean, reduce inoculum, and monitor for vector populations in both your strawberries and raspberries. And also, Perennia is now able to provide small fruit virus testing services for Nova Scotia growers. So that's everything for me.